Yersinia species consist upon a lot of organisms in this respective group, in which includes Yersinia pestis, from which are the actual disease plague arised, or it is even called Black Death. Apart from that, it has consist upon other genuses as well, which includes Yersinia enterocolitica, which infects the GIT, and the Yersinia pseudotuberculosis, which has been known to affect the lungs, causing some type of tuberculous type reaction. Now, apart from that, what happens is that it is a gram negative rod. Now, gram negative rod, this organism is zoonotic variety of gram negative rod, meaning that it has a specific reservoir which includes animals by which it is transferred to the humans. Another important thing about this is that it has low ID50, meaning that it has low infective index, meaning that even a few amount of organism are able to cause the disease in that respective human body. So, meaning that approximately small number of organisms can infect a human being. Other critical feature about this structure is that whenever it is stained, it goes under bipolar staining, meaning that the ends of the rod are being stained. This ends of the being rod by stain gives it a safety pin like appearance. Another thing apart from that is that it is surrounded by a polysaccharide capsule. Now, this polysaccharide capsule consists upon a few F1 antigen, which gives it a antiphagocytic property, meaning that whenever it is engulfed into the body, it cannot be digested. Apart from that, it also consists upon specific proteins, which is a V and a W protein. Now, this allows the organism to survive intracellularly. Whenever it is inside the cell, it will be not destroyed by these proteins. So what have we learned? It is a gram-negative rod, which has a bipolar staining, and it has a polysaccharide capsule, which has F1 capsular antigen, which has an antiphagocytic property. It has a V and W proteins, which allow it to survive intracellularly. We do not have a specific, it has an endotoxin and exotoxin, obviously because it is gram-negative rod, but its function are still unknown. Another important thing about this organism that it has outer proteins. Now, these outer proteins are known as YOPs. Now, this outer protein helps to inhibit phagocytosis by what it actually inhibits cytokine production by the macrophages. The macrophages produce a lot of cytokines. Now, what it happens is it shuts the trap of the macrophages. So, inflammatory reaction in the bodies are not seen. Now, this has protease that cleaves this is actually the protease that cleaves the signal transfer pathway, which inhibits the release of these cytokines. Its life cycle begins in, as we know, it is enzoonotic, meaning that it has animal origin. So this organism actually consists upon a specific vector. Now, this actually is transferred by fleas. Now, these fleas are carried upon certain rodents, which has rodent fleas, and certain in the United States, some specific species of dogs known as the prairie dogs were noticed to have some certain fleas that also carried this Yersinia pestis. Now, whenever this flea takes a blood meal of this organism, which is present in these animals, so the bacteria from the bacteria containing animal blood enters the flea. Now, the food is not ingested by the intestine or by the gastric system of the flea, because this organism consists upon a specific thick biofilm which does not prevent it to be ingested. So because of this biofilm, it gets stuck. So what does this mosquito does? It gags or it regurgitates. Now it regurgitates into a host. Now always know that humans are actually incidental host. It's supposed to infect some other dog or some other rodent but if it comes around a human and it bites them, all of this regurgitant bacteria is delivered to this human being and it enters the blood. Now, always know humans are the accidental host of this organism. Now, all of this bacteria then, when it enters the bloodstream, is drained by the regional lymph nodes. Now, what it gives rise to? It gives rise to your regional lymph nodes will be inflamed. It would be inflamed in such a way that it would be looking like a large bulge. Now, this large bulge is known as buboes, hence the name bubonic plague. It can also disseminate into the bloodstream and results in abscess formation anywhere part of your body. Another thing about this buboes is that if they are punctured, that if you think it's too big and you want to puncture it, you're actually releasing this organism into the environment and it can be carried by aerosol. So if this aerosol are carried by another human being, it would result in a condition known as mnemonic plague. Now, similarly, this disease would occur in the lungs of the another human being. Clinical features include that we will have a bubonic plague. Obviously, there will be a lot of buboes present on the body. They will be accompanied by fever, myalgia, and prostration, which are one of the very common symptoms of any sort of infection. This can be complicated and results in septic shock and even pneumonia. That was if the patient has inhaled the aerosol, that would result in bubonic type of plagues in the lungs. Lastly, what happens is if the patient is not promptly treated, this can result to death. It has 100% mortality if it is not being treated right. So the first thing what we need to do is we can, in order to assess the organism, we can make a clinical diagnosis of it by finding the bubonic plague. But some people actually, what they do is they take, in order to identify the organism more carefully, in order to rule out any different type of strains or antibiotic resistance which will later create. But it is very rare in this disease. So what we do is that we drain them. Now we drain a small amount. Not all of, we do not puncture it because it has still the ability to spread into the environment and become aerosol, which is quite dangerous because it can also give rise to a lot of community acquired or epidemic type of diseases. So after acquiring small pus and blood from the buboes, we do a Giemsa stain on it or a Weissen stain on it. We see a safety pin appearance, and in order to avoid such a dangerous condition, what we do is we do a fluorescent antibody staining, which also helps to detect the organism. Now, the treatment of such organism is giving the patient streptomycin or tetracycline. Tetracycline includes doxycycline, and always remember, if you know that this disease is being caused, you do not always have to drain it. The lymph nodes would eventually be de-infected and would decrease its size. So you do not have to puncture or drain all of the buboes at the same time.